Okay, okay. Class is already divided. No worries, we can start with our class. So in the today's class, like in the last lecture, we have finished our basics of cell cycle and cell division chapter. All you will remember what we
Yes,
have
ma'am.
done in the
Yes,
last.
ma'am.
Very well. So let's start today's class. So today we have the second lecture of cell cycle and cell division. In today's class, we will start with cell cycle interphase and little much basis of mitosis. Right. So we all very know what is the weightage of this chapter. In the last 2023, there are three questions that has been asked in cell cycle and cell division. In 2020, two questions, 21. So every on an average, two to three questions usually asked year to year. So can we have a quick recap what we have done in the last class? Ready for a quick recap?
Yes, ma'am.
Okay then. So let's start. Firstly, we have started with our topic with the chapter name. The chapter name is cell cycle and
And foundation.
cell division. So firstly, we will understand what is cell cycle. What we will done in cell cycle. In the cell cycle, we have studied there are two phases. The first one phase is
Interface.
L phase.
interphase, and the second one is
L phase.
M phase. Interphase is how much time period? It is of
2 years.
23
23
hours,
hours.
and M phase is for 1
1 hour.
hour.
1 hour.
There are- And there are three phases in the interphase. Name the three phases.
G1 phase,
G1,
S
G1
phase,
S,
and
phase,
G2.
S phase, G2 phase.
And there are four phases in the M phase. Named as
Prophase, metaphase,
prophase,
anaphase,
metaphase,
telophase.
anaphase, telophase.
very well done. Right. So in the G1 phase, what we have done?
No, ma'am, we have not studied yet.
I have not studied it. No worries. We will start with the G1 phase, S phase, and G2 phase in today's class. We all know human cell divide after how many hours?
24 hours.
24 hours.
And what about each cell?
90 minutes.
90
90
minutes.
minutes.
90 minutes.
3 minutes. Okay. Very well done. So let's start with today's topic. As you can see here in this animation, a cell is dividing. So firstly, we are saying a cell undergoes in a growth phase and then it get divided into two parts. Or we can say it gets split into two parts. Right or not? So we will understand this phenomenon in complete detail, how the cell is going into a growth phase and how the cell is going to divide into two parts. Clear? So let's start with the basic structure of a cell. In the cell center, we have a
Nucleus.
Nucleus.
nucleus, and outside environment is known as
Cytoplasm.
cytoplasm. And in the nucleus, we have a material which is known as,
Chromatin material.
Genetic material.
Chromatin.
can we call it DNA?
Yes.
Yes, ma'am.
Or we can call it genetic material.
Yes, ma'am.
Can we also call it chromatin?
RNA.
Yes, ma'am.
Can we also call it chromosome?
Yes, ma'am.
Yes, ma'am.
What are the difference between these different terms? What is the difference between DNA, chromatin, and chromosome? We all know DNA is genetic material.
Yes, ma'am.
Then what is chromatin and chromosome? Are there any new structure or anything else?
No, ma'am. Chromatin, it is a form of DNA which is coiled in the form of a thread, and chromosome, it is a also form of DNA which is uh, means given a proper shape like a chromosome which consists P arm, Q arm, and centromere.
Very well done. Let's have a quick recap of DNA. We can see this is a DNA, double-stranded DNA. Can we predict the length of DNA?
2.2 meter.
2.2 meter.
Very well. 2.2 meter. And do we have any idea about the size of a nucleus?
1 into 10 raised to minus 6.
6.6 into 10 to the power minus 6 meter. So we need to fix this 2.2 meter DNA into a nucleus which is of size 6.6 into 10 to the power minus 6 meter. Is it possible or not? How we will
Yes,
make it possible?
in the form of chromatin condensed structure.
We will make it condensed, right or not? So how I will make it condensed? I'll wrap it into a condensed form. Then this condensed form of DNA is known as
Chromatin material.
Chromatin.
chromatin material. Then in the form of chromatin material, in
Good
the form
evening,
of
ma'am.
chromatin material, all genetic material get easily incorporated into the nucleus, which is of size 6.6 into 10 to the power minus 6. Right?
Yes, ma'am.
Yes,
Then
ma'am.
chromatin is the most condensed form. Then why we are needing to convert chromatin into chromosome? What is the need of chromosome here? Why we are converting
For
chromatin into chromosome?
Because we want to divide it into two nucleus.
Very good, Tamanna. Tamanna, you have already studied this now in my different batch.
No, ma'am, because I joined late in class tomorrow.
Okay, okay. So chromosome is also a condensed form of DNA. Chromatin and chromosome both are the
condensed form of DNA.
condensed form of DNA. The chromatin is in the form in which we cannot split genetic material into two equal parts. If someone says me, let's divide the chromatin material into two equal parts. Is it possible in the form of chromatin material?
No, ma'am.
It's not possible, na? We cannot divide it into two equal parts. But if I convert it into a X-shaped structure, which is known as chromosome, then in this form, I can divide it into two equal parts easily. By splitting the two arms, by splitting it from here, I can divide it into two equal parts. Are you getting my point? What is the
Yes,
need
ma'am.
of,
Yes,
what is
ma'am.
the need of chromosome here? We will convert chromatin into chromosome at the time of division. At
Division.
the time of division, we will convert chromatin into chromosome. So in this case, we can ask a question here. Let's write a question. In a dividing cell, genetic material will be in the form of
Chromosome.
chromosome.
Chromosomes.
Very well. And if someone asks, in a non-dividing cell, genetic material will be in the form of
Chromatin material.
Chromatin.
chromatin material. So now we are clear with both the terms, chromatin as well as chromosome. Clear? Okay then. Let's move on to the next point. Have a cell. Okay? And we need to divide it into two equal parts. Before division, या फिर we can say before dividing the cell, we need to increase the cytoplasmic content of our cell, as well as we need to increase the genetic material of a cell so that we can divide it into two equal parts. Before dividing a cell, we need to divide. We need a process in which cell can grow itself. And this process will occur in which phase? Phase.
Interphase.
Is also known as, this phase is also known as preparatory phase, because cell will prepare itself,
Resting phase.
and it is also known as resting phase. Why we are calling it resting phase? Because in this phase cell will not divide itself. Cell will not divide. Cell will only prepare itself for the division. Cell will prepare itself for a proper division. So the first phase of interphase is, we can make a chart here. The first one phase we have is G1 phase. G1 phase is also known as gap 1.
Gap 1 phase.
Very good. And the second phase we have is
S phase. Synthesis
S
phase.
phase.
S phase. S represent here synthesis phase. And then we have
G2 phase.
G2 phase.
phase.
Capsule phase.
Phase is also known as gap 2 phase. Okay, we will write down all the points here. In the G1 phase, what will happen? G1 phase may growth occurs. And
Continuous.
growth in terms of
Cytoplasmic increase.
cytoplasmic increase. And we have already studied this. Cytoplasmic increase is a continuous process now.
Yes.
Yes, ma'am.
Continuous process. तो सेल का यहाँ पे क्या होने वाला है? Cell will, in this phase, growth will occur in the terms of cytoplasmic increase. And cell is spending here how much time? Out of the 23 hours. Out of the 23 hours, how many hours will spend here?
8 to 12 hours.
12 hours. And in S phase, it will spend how many hours?
6 to 8 hours.
6 to 8 hours. And in G2 phase,
3 to 4 hours.
3 to 4 hours. So the in which phase cell is spending maximum time?
G1 phase.
G1 phase. If cell is spending maximum time in G1 phase, then there is more probability to have any damage in this phase only.
Yes.
तो सेल यहाँ पे maximum time spend कर रहा है, so there are more chances to have any type of damage in this phase only. So if any type of damage occurs, then the repairing of that damage will also occur in this phase.
Yes, ma'am.
Damage हुआ तो then also repairing will also occur in this phase, na? So we can write this point here.
Yes, ma'am.
Spending maximum time occurs. Clear?
Yes, ma'am.
Very good. And in the S phase, what will happen? DNA
Replicate.
content
Gets doubled.
will, yeah, so we can write it, get doubles. But number of chromosomes?
Is same.
Very good.
Remains same.
Number of chromosome remain same. Let's see. If DNA is getting doubled, then the in the last phase, in the last slide, we have studied ki we need a condensed form of DNA to pack it into the nucleus. Now we have 2.2 meter DNA, but after this phase, DNA will become 4.4 meter now.
Yes, ma'am.
So we will be needed more type of packaging.
Yes, ma'am.
Yes, ma'am.
And हम DNA को we usually packed DNA with which structure? We packed DNA with histone proteins. We packed DNA with which protein? Histone protein.
Histone protein.
And how we packed it? DNA is negatively charged and histone proteins are positively charged. So there will be a negative positive attraction. So DNA will get wrap itself on the histone protein. DNA is negatively charged, histone is positively charged, as well as DNA is acidic in nature, and histone proteins are basic in nature. So there is a opposite attraction between DNA and histone protein. So DNA will pack itself with the help of which protein? Histone protein. So in the S phase, if we are getting double DNA content, then we will be needed more histone proteins.
Yes, ma'am.
So it means histone proteins will also double here.
Yes.
Yes, ma'am.
Histone protein double itself. All 3 points are clear.
Yes, ma'am.
Next,
Ma'am.
also centriole also get double. We have studied centriole in the last to last chapter. Cell structure. In the cell
Yes,
structure or
ma'am.
the cell, the unit of life. Centriole. What is the use of centriole?
Cell division.
Centriole helps in cell division. So in this phase, in S phase, centriole will also double itself. Centriole can be found in which cell? Animal cell or plant cell?
Animal cell.
Animal
In
cells.
animal cell only. So it means centriole will get double in animal cell only.
Yes, ma'am.
All points are clear?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Very good. And then what will happen in the G2 phase? Protein,
It is synthesized by tubulin protein.
protein synthesis. And which protein? Tubulin protein.
Tubulin protein.
Tubulin protein helps in formation of spindle fibers.
Microtom tubal.
स्पाइंडल फाइबर्स बनाने में ये हेल्प करता है. Are we clear with all 3 phases? The first one in G1 phase. In the G1 phase, cytoplasmic growth occurs. A cell spend 8 to 12 hours. It spend maximum time in this phase. So if any kind of damage occur, then the repairing will also done in this phase. Then after finishing the G1 phase, cell will enter into S phase, where DNA content will double itself. If we are doubling the amount of DNA, then we will need it more histone protein. Then histone protein will also double itself. Then centriole will also double itself, but in plant cell only. Number of chromosomes remains same. In G2 phase, protein synthesis occur. Protein, which protein? Tubulin protein, which help in the formation of spindle fibers. As well as we have two semi-autonomous organelles. Which are semi-autonomous organelles? Mitochondria and chloroplast. Mitochondria and chloroplast. These are the two semi-autonomous organelles, na?
Yes, ma'am.
Yes, ma'am.
In this phase, mitochondria and chloroplast also double itself. लिख लो इसको जल्दी से. Write down all these points, then we'll move on next. Done?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes,
Okay,
ma'am.
then. Let's move on to the next point. Yeah. Let's understand this. Firstly, we have a cell. Okay? And cell is going into firstly G1 phase. After finishing the G1 phase, cell will enter into in which phase?
Yes.
S phase.
S phase.
S phase. ठीक है, बट बिफोर एंटरिंग इनटू द एस फेज, ध्यान से सुनना है, बिफोर एंटरिंग इनटू द एस फेज cell has to clear some checkpoints, or we can say here S phase has some checkpoints. If cell will clear all those checkpoints, then cell can easily enter into S phase. What are those checkpoints? The first one.
G not
हाँ,
phase.
नहीं, आई एम सेइंग कि जो हमारा S phase है, S phase has some checkpoints. If cell has cleared all those checkpoints, then cell can easily enter into S4. The first one checkpoint is cell should have sufficient food. Cell should have sufficient food. And if cell has not sufficient food, then what will happen? Cell will
It
not
will.
be cell will not be able into S phase. Cell हमारी S phase में enter नहीं हो पाएगी. Second checkpoint, there should be no damage. And if any type of damage found, then cell will not be able to enter into S phase. हुए ना checkpoints?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
And the third one condition, once a cell enter into S phase, it has to divide. What does it mean? Once a cell enter into S phase, it has to divide. It has no other option to get back into a G1 phase. The first one point, cell should have sufficient food. Cells, there should not be any type of damage in the cell. And if a cell once enter into S phase, then it has to divide. There is no option to get back into G1 phase. Okay? So if cell clarify all these checkpoints, then cell can easily enter into S phase. But if there is no sufficient food or there is any type of damage found, then what will happen with cell?
It cannot enter into the X phase.
It cannot enter into S phase. Then where cell will go?
G0 phase.
There is one option, cell can enter into G0 phase. Another one option. We've already studied meristematic tissues and permanent tissues. Can someone tell me the definition of a permanent tissue? What are permanent tissues? Permanent tissues are those tissues which never divide. And meristematic tissues are those tissues which continuously divide throughout their life. Right, Mishpa?
Yes, ma'am.
If cell has no sufficient food, then cell can permanent itself. Cell वहीं पे permanent हो जाएगा. अगर cell के पास
Yes.
sufficient food नहीं है, unless cell can enter into G0
S
phase.
phase.
No.
That phase.
अगर sufficient food नहीं है, then it cannot be able to enter into S phase. वो S phase में enter नहीं हो पाएगी. Cell will enter into G0 phase.
G0 phase.
G
So
not
we have
phase.
two conditions. We have two conditions here. Cell become permanent. Cell permanent हो जाएगा. And the second one, cell
Temporary.
enter into G0
G0
phase.
phase.
G not phase.
Very good. And permanent होने के बाद क्या करेगा? What will happen with cell in the permanent case?
It will not divide.
Role fix हो जाएगा उसका. Cell will perform a specific function. It will not divide. वो divide नहीं करेगा. And after performing फंक्शन, अपना स्पेसिफिक फंक्शन परफॉर्म करने के बाद सेल विल डाई, natural death of cell. But when cell enter into G naught phase, again it will not divide. कब तक डिवाइड नहीं करेगा? Until asked to do. So it means in this phase, cell is metabolically active ना?
Yes.
Yes, ma'am.
Yes, ma'am.
Cell is metabolically active. तो it means cell क्या कर रहा है? Cell exit कर रहा है. Cell exit cell cycle for short period. For a short period. कुछ टाइम के लिए, for some time, cell is getting away itself from cell cycle. But cell can divide again if we ask it to divide. Clear? In which case cell will become
Yes,
permanent,
ma'am.
in which case cell will enter into G naught phase. And G naught phase is also known as quiescent phase. G naught phase occurs after which phase? G
G1
naught
phase.
phase. G1 phase.
G1 phase.
And G naught phase occurs after G1 phase. ठीक है, ये भी क्वेश्चन नीट में आपका पूछा गया है. क्लियर यहां तक?
Yes,
Yes,
ma'am.
ma'am.
So we are done with our S phase, G1 phase and G2 phase. So after finishing the G2 phase, cell will enter into which phase? Ajit,
M
can
phase.
firstly let me do. Can we solve some questions? Let's read this question. And you can answer in the chat section. Read this question. This question is already asked in NEET 2020. Very good, Mishwa. Very good, Devisha, Anushka, Anusha. Identify the correct statement with regard to G1 phase. Cell is metabolically active, cell will grow, but it does not replicate its DNA. Very well done, very well done. So the correct answer is C. Let's move on to the next question. In S phase of the cell cycle, what will happen? This question is already asked in 2014. Amount of DNA doubles in each cell, amount of DNA remains same, chromosome number increase या फिर amount of DNA is reduced. Very good, very good. जल्दी से आंसर करो, हरिए बच्चों. But in the chat section, Ramesh sir has shared a attendance form with you. I'm sharing it again. तो आप लोग एक बार ना इसको fill कर देना, ठीक है? Just after finishing the class.
Yes, ma'am.
Done na? So the correct answer is A. Next question, very easy question. And asked in 2019. Very good, Harshada. Very good, Tripali. The correct sequence of phases of cell cycle is the first one is G1, then S, then G2, and then Very well done. Very well done. The correct is A. Next question. We are saying the correct answer is B. Okay, let's see the correct is B or not. Some dividing cell exit the cell cycle and enter into a vegetative and active state. This is called quiescent stage, which is G naught. And this process occurs at the end of G1 phase. Very nice. Okay then. So we are done with our question. I hope ये वाला topic आप सभी लोगों को clear हुआ होगा. You all are done with topic. So
Yes,
can we?
ma'am.
Okay then. So now we will start with a new topic, which is your mitosis. Clear? Let's start with mitosis. We all know mitosis is also known as
Equational division.
equational division. Very good. And we'll also call it most dramatic period of the cell cycle. Most dramatic period of cell cycle. So my first question is, why we are calling it equational division? Just raise your hand if
Chromosome
you can.
replicate.
Raise your hand. Yes, Anusha.
Chromosome replicate and get equally distributed into two daughter cells.
Very good. Anyone else? Yes, Mishpa. Mishpa का left हो गया. Harshada, do you want to try?
Ma'am, two N chromosomes get converted into haploid chromosomes.
No, this is known as reductional division. Yes,
Nuclear division, ma'am.
Gayatri. Do you want to try?
Ma'am, the number of chromosomes in the parent and the daughter cell remains same, so it is called equivalent division.
Exactly. We will call it equational division. In the equational division, the number of chromosome in the parent cell and in the progeny cell will remain same. But if number get reduced to half, then it is known as meiosis, reductional division. This is the basic difference between mitosis and meiosis. Mitosis is known as equational division and meiosis is known as reductional division. Right, हर्षदा? समझ आया आप कहाँ पे मिस्टेक कर रहे थे?
Yes, ma'am.
Okay then. Chalo, let's start. So we can write this point here. Why we are calling it equational division? Because number of chromosomes in parent and progeny cell remain same. Also, in the mitosis, we have 4 phases. So we'll start with all 4 phases in a tabular form. The first one phase we have
Prophase.
Prophase.
prophase. Next,
Metaphase.
next,
Anaphase.
Anaphase.
next,
Telophase.
Telophase.
telophase. Okay. Prophase is known as the longest phase. Prophase को हम बोलते हैं longest phase of the mitosis. And the shortest phase is
Anaphase.
Anaphase. Very well done. In the prophase, what will happen? We'll write it step by step. The first one step is in the prophase, DNA start condensation. Why it is getting condensed? Why it is getting condensed? Because mitosis is a division phase and interphase is a non-division phase. So DNA will convert itself from chromatin to
Chromosome.
Chromosomes.
chromosome. That's why the first step should be the condensation of DNA, na?
Yes,
Yes, ma'am.
ma'am.
Gayatri, any doubt?
No, ma'am.
Okay, so the DNA will start condensation. It will convert into chromosome. The second step is centrioles moves to opposite poles. Centrioles start moving सेल के opposite poles में and start forming spindle tubules, or we can say spindle
Fibers.
fibers बनाना start कर देंगे, या फिर we can call it micro tubules.
Tubules.
ठीक है. The next one is endoplasmic reticulum, nucleolus and
Golgi
Golgi
complex.
apparatus start
Disappearing.
disappearing. And the last, nuclear envelope
Breaks into vesicle.
nuclear envelope also start disappearing. Start disappearing. ठीक है. Metaphase. How we will identify the cell enters into metaphase? How we will identify cell has entered into metaphase?
There is disintegration of nuclear envelope.
Exactly. Complete disintegration of nuclear envelope. Complete next point, completion of condensation of chromosome. Also finished here. Completion of
Condensation.
condense chromosome. And metaphase is also known as best phase to
To
study
study the morphology of chromosomes.
morphology. Very good. Best phase to study morphology. Because this is the phase in which chromosome is mostly condensed. This is the phase in which chromosome is mostly condensed. And this is the only phase in which we can actually count the number of chromosome. How many number of chromosome found in human cell?
46.
23.
So we can say it's 23 pair, or we can say it 46. So we have counted these 46 chromosome in which phase?
Metaphase.
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 Right. And in metaphase only, all chromosomes itself at where metaphase split. सारे के सारे क्रोमोसोम अलाइन कर लेते हैं अपने आप को कहां पे? मेटाफेज प्लेट पे, एंड गेट अटैच्ड विद
Kinetochore.
kinetochore, and from kinetochore to spindle fiber. Take diagrams also in the next slide. ठीक है. Next, in the anaphase, what will happen? This is the shortest phase. So in this phase only, centromere split and chromatids
Separate.
moves opposite poles. And telophase is just opposite to the prophase. Write down it itself only. टेलोफेज में क्या होगा? इन द प्रोफेज डीएनए स्टार्ट कंडेंसेशन, डीएनए कंडेंसेशन स्टार्ट, बट इन टेलोफेज
Decondensation.
decondensation. Very good. In the telophase, decondensation. Is endoplasmic reticulum, nucleolus, Golgi apparatus all start disappearing. Then is phase start
Reappear.
reappearing. And chromosome lose its identity. Why they lose its identity? Because chromosomes get converted into chromatin material. है ना? तो chromosome lose its identity, and the last should be nuclear membrane, nuclear envelope reappears. लिखो इन सभी पॉइंट्स को, then we will make some diagrams. Let me know with a thumbs up when you are done with writing process. Okay then. So let's start making some beautiful diagrams. Try to make diagrams on your own. Let's see. This is our nucleus. In the nucleus, we have genetic material in the form of chromatin, and the outside is
Cytoplasm.
cytoplasm. Then in the second phase, what will happen? Nuclear membrane abhi as it is hai, and it start condensation. So we can call it early prophase. We can call it early prophase. There is mitochondria. There is Golgi apparatus. Golgi apparatus भी यहाँ पे है. There is endoplasmic reticulum also. ठीक है? And in the next phase, what will happen? पूरा का पूरा nuclear envelope disintegrate होना start हो चुका है. We have chromosomes here, and these organelles also disappear. We can call it late prophase. And in this phase, chromosomes. Which phase is this?
Metaphase.
Metaphase.
Have centriole. Centriole also start moving on opposite poles. Now we have centrioles on opposite poles. And they start forming spinal tubules,
Spindle fibers.
spinal fiber. And spinal fiber get attached to
Kinetochore.
kinetochore. This is metaphase. ठीक है? And what will happen in late metaphase? इसको अच्छे से देखना.
They are converted
Chromosomes
into
are
types.
aligned.
In the center, we have centromere, and at centromere, we have a disc-shaped structure. The disc-shaped structure is kinetochore. It is made up of protein, na?
Yes, ma'am.
Very good. And there is we have centriole, and look at this. ऐसे ही कुछ दिखेगा ना हम लोगों को.
Yes, ma'am.
एक क्रोमोसोम है ये हमारे पास, ठीक है. क्रोमोसोम की इनको क्या बोलते हैं? These are known as chromatids. These are known as chromatid. So in a single chromosome, one chromatid is attached with one pole and another chromatid is attached to another pole, na?
Yes, ma'am.
One chromatid is attached with one pole spindle tubule and another chromatid is attached with another pole spindle tubule. So both will attract the chromatid toward itself. Then what will happen?
Monad will get diet will get converted into monad, and both will start moving to the opposite poles.
Centromere gets split. Centromere gets split, na? If
Yes.
centromere gets split, then we will look it like this. Like chromosomes are trailing behind centromere. Centromere. Chromosomes are trailing behind centromere.
Centromere.
Then which phase is this?
Anaphase.
Anaphase.
Anaphase. What happened in anaphase? The first centromere split and chromatid move to the opposite.
Opposite poles.
Very good. And in the next phase?
Decondensation.
Decondensations. Chromosomes start decondensation. Then they will look like this. And in the next phase, what will happen? There will be a nuclear envelope again. And all cell organelles will also start reappearing. Then this phase is known as telophase.
Interface.
Done. And this plate is known as this plate where all chromosomes are aligned.
Metaphase plate.
Metaphase plate. This is equator. जैसे होता है ना ग्लोब में we have a equator and we have a pole. When we move on a polar side, जब हम पोल की साइड मूव करता है तो वो क्या होता है?
Equator.
All live on equator side. That's why there is moderate temperature. अच्छा सारे डायग्राम्स क्लियर है ना? All diagrams are clear to all?
Yes, ma'am.
Yes, ma'am.
जैसे एक बार हैंड रेज करके अपना अटेंडेंस मार्क करा लो. एंड जिसको हिंदी बिल्कुल नहीं आता था, अच्छा मुझे बताओगे कौन आपने कोई नए वर्ड सीखे हिंदी के? डिड यू लर्न एनी वर्ड्स ऑफ हिंदी? So first class में I told you ना we learn different languages. Gayatri, are you able to understand some words of Hindi?
Ma'am, little bit, ma'am.
Little bit. Let's see. हमारा जैसे classes when we'll further continue these classes around 2 months, you will be able to understand Hindi. So many words.
Okay, ma'am.
ठीक है? So try to speak in Hindi also. If you learned any word.
Okay, ma'am.
Okay. So we have done with this telophase. Now we can start with next one. Let me check the timing. No, we don't. Resend the link of it. I'm sending it. I'm sending it. Don't worry. Don't worry. So can we summarize today's learning? What we have learned in today's class? We have learned all phases of mitosis. We have
All
learned
phases of
different
interphase.
phases of mitosis. The first one is PMAT. What is the PMAT? Prophase, metaphase, anaphase, telophase. In in which out of the four phases, in which phase centromere split?
Anaphase.
Anaphase. And in which phase all chromosomes get aligned to a equator, and that equator is known
Metaphase.
Anaphase.
and that equator is known as where all chromosomes get aligned.
Method.
Metaphase.
Metaphase plate.
Metaphase plate. Very good. And which phase is known as the best phase to study morphology of a chromosome?
Metaphase.
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 Very good. Which phases? With write the name of two phases which are opposite to each other.
Prophase and
Prophase and telophase.
Prophase. Telophase.
Prophase and telophase. And just have a look to this point also. Ki all these phases occurs only in eukaryotes. Interphase, prophase, metaphase, anaphase, these phases exist only in eukaryotes. If someone asks you a questions regarding prokaryotes are bacteria na? We have learned in biological classification chapter. So there is no such type of phases in case of prokaryotes.
Prokaryotes.
प्रोकैरियोट्स के अंदर कोई phases नहीं होता है. Prokaryotes only divisions happen, like binary fission. We have learned in 10th class binary fission, multiple fission. So in prokaryotes only fission occur. There is no mitosis, meiosis happens. ठीक है? Okay then. So now homework time will share DPP in your group, as I have already shared a MTG book with you. Did you find it in your group?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
I have shared a drive link with all of you, and you can paste it in somewhere else. I am just checking the PDF. There is a PDF. Yeah, I've shared. You can find it. उस PDF में आप check करेंगे. You can go with the chapter cell cycle and cell division. There you can find topic wise question. So everyone will solve all PYQs. Latest year PYQs. Latest means 2005 plus. Only 2005 plus questions you will solve from that PDF only. Okay, that will be your homework. And before coming to the next class, because in the next class we will finish this chapter. Next class में हम ये chapter finish कर देंगे. In the next class we will start with cytokinesis and then we will go with meiosis. ठीक है?
Okay, ma'am.
Okay,
Okay.
then.
Yes,
And
ma'am.
additional resources you can refer. I have already shared MTG book with you. Also, you can refer Dr. Ali MTG, Dr. Ali Objective Biology, and your NCRT book. NCRT is must. You cannot ignore NCRT. And in the next class, we will study meiosis and little phases of cytokinesis also. So, have you all filled the Google form for attendance? Yeah, Namrata, I'm just sharing it. Mark your attendance, everyone. If you are done with your attendance, then you can leave the class. We will start with next topic in the next class. And